Chemistry is all fine and good, but what separates life from non-life is the cell. You are made of trillions of these things, all working and doing things to keep your sorry butt alive. Right now, there are over 300 cell types in your body, including skin cells, red blood cells, bone cells, neurons, nephrons, morons, lymphocytes, and those found in ciliated pseudostratified columnar epithelium. Okay, now I'm showing off, and if you're wondering if I said morons, I did. Okay, so what are cells? These are compartmentalized living structures that carry out various functions. The plasma membrane separates the interior from the exterior of the cell, and the structure, function, and even species assignment is all dictated by DNA found in the nucleus, well, of some cells. Cells that work together to carry out a similar function make tissues, and in turn, multiple tissues types that work together form an organ. And those organs that work together to carry out a major biological process are called organ systems. And finally, these organ systems make up an organism. There is a problem. Unless you are an egg or multinucleated organism made up of a single cell, such as this algae, almost all cells are too small to be seen without the aid of this magnificent tool invented shortly after the telescope,
the microscope. Before beginning, let's learn how these magnificent tools help us see the invisible world.
So just how small are cells? If you take the size of a single red blood cell, it measures 8 millionths of a meter, or 8 micrometers. To give you the sense of how big a micrometer is, take a meter stick and note that the small dashes are millimeters. From there, 1,000 micrometers will fit in 1 millimeter. In other words, if you take 125 red blood cells lined up end to end, it will fit into the space of a single millimeter. It is that small. Microscopes work by magnifying objects so our eyes can see the unseen by using two specially shaped lenses and mirrors to project the image into our eyes. Because of the way light travels through these lenses, specimens that appear right side up to us will be upside down under the microscope. Most people at some point will use a light microscope, which utilizes light to help you visualize a specimen. They are relatively cheaper than other types of microscopes and used most popularly in educational settings. The advantage of light microscopes is that it can allow you to see both living and non-living organisms. But since most cells are transparent in color, stain must be added to see them, which unfortunately kills the cells. The stain improves the contrast of the specimen since a living, clear-colored cell has a lighter contrast. The microscopes I use in my classrooms can magnify from 40 to 1,000x. That means we can view specimens 1,000 times closer than the naked eye. Magnification is the degree of enlargement of an object. Another type of microscope used in labs and educational facilities is a dissecting microscope. The magnification is much lower at 20 to 80 times, but it provides the advantage of seeing three-dimensional objects. Dissecting scopes are useful in seeing larger organisms in much greater detail. For example, look at my hand under the microscope. You can see the intricate folds and hairs, and I really need to moisturize. The last type of microscope is one that I have actually never used. That's because it is limited to research facilities due to its expense and large size. These are called electron microscopes, and they work by using a detailed three-dimensional image of cell surface structures and organelles not seen with a light microscope. Notice this Salmonella bacteria under a light microscope versus the same organism in an electron microscope. Not only is Salmonella much larger, but appears more three-dimensional. Unfortunately, due to the imaging process taking place in a vacuum and use of special coating materials on the cell, living specimens cannot be viewed with an electron microscope.
Microscopy has come a long way since the time of Dutch shopkeeper and drapery maker Antony van Leeuwenhoek, who in the 1600s designed a special craft lens. Although crude in design, he was able to see movements of an organism that we now know as Giardia lamblia. And well, that sucked for him because the disease-causing protozoan parasite causes horrible gastrointestinal diarrhea. Ugh. Isn't it funny how a bad bout of the helps springboard our understanding of cell biology? But where did the name cell come from? In 1665, scientist Robert Hooke developed the name cell from the Latin word for small room, cella. He called them cells because the cork tissue he viewed under the microscope looked like tiny rooms. In the 1830s, botanist Matthias Schleiden and zoologist Theodor Schwann developed the unified cell theory, which is still applicable today. Cell theory has three components. All living things are composed of one or more cells. Cells are the basic unit of living things, and that all new cells arise from existing cells. Thus far, cells fall into two broad categories, prokaryotic and eukaryotic. The main difference lies on whether the cell has a nucleus or does not. Prokaryotic means before the nucleus, or cells that existed before the presence of nucleated cells. Eukaryotic means true nucleus, or organisms that are made of nucleated cells that house their DNA. And before delving into the differences between prokaryotic and eukaryotic cells, we need to look at what all cells have in common. And to do this, we must shrink down to the level of a cell, just like we did with the atom. I know this is cringe, but let's go!
Anyone there? No, just kidding. I'm done filming myself for a bit. It takes a lot of time to set all this up, where if I just speak into a microphone, it's much easier. Anyway, all cells have a plasma membrane, which is a barrier surrounding the cell as well as other cellular components, and this dictates what enters and exits the cell while separating the internal mechanisms of the cell from the external environment. The cytoplasm is a jelly-like region within the cell that contains cellular components. The chemical makeup of the cytoplasm can differ significantly from the outside of the cell, which is another reason to have a cell membrane. All cells have DNA, which contain the genetic material of the cell, and finally, all cells have ribosomes, which take genetic material in the form of messenger RNA to help make proteins. And some, DNA houses instructions for making proteins, which are then copied onto messenger RNA and made into proteins by ribosomes. And now we will delve into the most ancient life forms, the prokaryotes. Prokaryotic cells are the cell type that makes up the two life domains, bacteria and archaea. You probably already are familiar with bacteria, but archaea are superficially similar in appearance, but vary widely in genetic material. Both domains of life harbor prokaryotic cells, which are much smaller than eukaryotic cells and lack a nucleus. Instead, the DNA is condensed into an area called the nucleoid. In addition to a plasma membrane, prokaryotic cells contain a cell wall, and while some eukaryotic cell groups also have cell walls, the chemical composition of prokaryotic cell walls differ greatly. The cell wall helps to protect the cell, preventing from dehydration and provide the organism with its shape. Some bacteria, in addition, have a capsule outside of the cell wall, which helps the organism attach to surfaces. In addition, some prokaryotic cells have a flagella used for locomotion and a pilus used for a special type of gene exchange between bacterial cells called conjugation. Eukaryotic cells are much larger than prokaryotic cells, and they have a membrane-bound nucleus that houses DNA. In addition, there are many membrane-bound structures inside of the cell called organelles. And like body organs, organelles are specific compartments that carry out specialized tasks, but we'll look more at these later in greater depth in the next video.
As mentioned before, cells are really small, and while eukaryotic cells range from 10 to 100 micrometers, prokaryotic cells range from 0.1 to 5 micrometers. So why are cells so small? You see, smaller sizes allow for the quick uptake, release, and distribution of chemical components produced and needed by cells. If cells were larger, it would be less energy efficient and more time consuming for cells to carry out basic functions. And now that we know more about prokaryotic cells and why cells are small, as well as being introduced to eukaryotic cells, let's learn more in detail about eukaryotic cells in our next video.